Okay, so great. So we finished going over the basics, right, of many of the things that are the many things that are relevant for in the context of brain network analysis. And now we are going to go over a particular case study, which is some recent work that myself, Shitabra, myself, and Anand, who will be talking tomorrow. So some work that we have done on the macaque brain. I had initially planned this to be 2 hours, but I think I might be able to do this in an hour and a half. Let's just see how it goes. Okay, we'll do our best. Okay, so but this is why you this is what happens when you make slides when you are on cough syrup medication. Okay, so let's talk about the primate. Okay, um, people have been studying the primate brain for a long time because of amongst several model organisms. It's the closest in terms of several of its features to the human brain, and so for a lot of treatments and various other things, people study the macaque brain. Well, not just the macaque, the primate brain. And so as you can imagine, there's a lot of data available for the connectivity between different regions of the brain. The macaque is one of the model species that has been most widely studied. And there was an initiative all the way back in the late 90s by Rolf Kotter, which was to sort of create a database which has which kind of brings together like all the information that which up to that point people knew from published studies about the connectivity data of the macaque. Okay, so this was essentially taken from track tracing studies, right? So basically, so you have the way the experiments are done is they put some sort of tracer in a certain location, and then it sort of goes along the axonal tracks, and you can sort of see like what are the regions that connect to which other regions. So this is done over like very long period of different studies done over a long period of time, different protocols and so on. So there is no sort of so it's a very sort of mixed bag in terms of the consistency of data, right? But at least it's a start. I mean, it's not like there's one experiment where they did everything. I mean, that would be invaluable, but this is pretty good, right? So this is done over like hundreds of such axonal track tracing studies, and so this gives a fairly comprehensive record of the wiring of this brain. And so the reason why these track tracing studies are they can be trusted is because it gives like very kind of incontrovertible evidence of long range connections, right? And so and also the direction, which is also important, like which is the direction in which the tracer is sort of moving and so on. That also gives you some useful information, right? So so this COCOMAC database was something that was I think now I'm not entirely sure. It seems to be sort of not maintained nowadays. I'm not entirely sure about. I don't know whether people are still adding to it. I know that the website is still up, or at least one version of the website is up. The original website I think is down, but they've got a new version of the website. But I don't think it's been updated for a very long time. But I'm not going to specifically talk about the COCOMAC. The reason why I'm mentioning the COCOMAC is because there was a work that was done by Modan Singh back in 2010, where they said, all right, so we have this data available to us, has these hundreds of experiments, and what we should do is we should collate this even further. Just boil this down into just like a single network, right? A single network that tells us all the connections that exist between the different brain regions of the macaque. Okay, And that is precisely what they did in this paper over here. So as you can see, so these are like these boxes, they represent the different brain regions, and these are all the various links which are obtained from the studies. As you can see, going around the circle, these are the different parts of the brain: temporal lobe, occipital lobe, and so on. And there is something else which is going to be an issue which we're going to have to look at, which is that going from outward, from inward to outward. So right in the middle, you notice something. It says BR, right? So basically, all of these have abbreviations, right? So they have CG number 2, SPL, and so on. All of these are just abbreviations for different brain regions. So, by the way, this is actually a very interesting paper. I mean, there's way too much to go into right now, but I strongly suggest that you check it out just to see the methodology that they used. But there is one problem, at least from our end, which is that BR actually stands for brain. So that is okay, fine. So you have a node which is the brain, but then you also have some other nodes as well, like CX is the cortex, right? Now this becomes slightly problematic. So now you start to have various and this is the diencephalon and so on. Basically what this is telling you is okay, this is a region, these are its subregions, these are its subregions, and so on, right? So really the only regions that have no further subdivisions are the regions on the outer sort of circle over here. So these regions, they don't have any further subdivisions, right? And some of these regions in the middle, they don't have any subdivisions as well. I mean, that's but many of them do, right? But so why is this a problem? So okay, so this is just from there that this looking. Anyway, this is not too bad. So this is from that table. You can see that these are, this is, so they have a long table in their supplementary material where they mention the details of all the various regions. Yeah, this is the brain and the diencephalon, hypothalamus, thalamus. So the thalamus is a node in the network, and then you say these are the sub, and so a level corresponds to well, they call it hierarchical. This is not the same hierarchy that I spoke about last time. Please keep note. So when they say hierarchy, they mean hierarchy in terms of one being contained within another. So this is very different from what I was talking about. So this level 0 is obviously the entire brain. Level 1 is the diencephalon, 2 is the hypothalamus, and so thalamus, you can see thalamus has a further level 3, right? And this has a further level 4, and so on. And then you go to the next 3. So this is how it's ordered. Okay, so what's the problem with this? Okay, so you say, okay, fine, cool, they have all these. What's the problem? The problem is, like, suppose you have a situation like this, right? So let's just say I have region A, region B, and region C. And now I know from my data from the more than seeing network that you have a connection going from A to C, okay? And you also know that there's a connection going from B to C. Addition, as I said, B we know is a subset of A. It's a lower hierarchical level of A. So if you look at on this chart over here, so let's just say that you see this number 5 you have over here. So you have this CIM. So CIM, this is a subset of I guess it's C number 4, which is in turn a subset of ML, and so on. So you know that these are, this is where they lie. So what's the problem? The problem is, how do I know that this link and this link are not the same? Because you see, these are not taken from the same experiment. These are taken from different experiments done at different times, different labs, different protocols, and so on. Unless I go and manually check every single experiment and look through every single record of how the details, I'm not going to know for sure whether it's possible that at the time when they're doing the experiment to get the link from A to C, that they just okay, I'm not interested in looking at B. I just want to know from A to C what is there a link, and so I found it, right? But later on, maybe possibly they said, okay, I'm going to focus on somebody else. I'm focus on B, and I found a link to C. It just could be that these are the same links, right? There's no way of knowing for sure, right? So these kind of redundancies can happen. So I could have multiple links that essentially tell me the same information, right? That's a problem. And there's also another problem, which is that I mean, the very fact that I have parent layers, it becomes ambiguous as to how to interpret the result, because the parent layers contain its children as well, right? So any inference that I'm going to be making, it's going to be different, because these are not like distinct units. These are units which contain other units, right? It would be much easier if I'm working with distinct units, right? So for this purpose, what so Anand, myself, and Chitavra, what we decided was that, all right, so this is the original network of Modan Singh, right? So in their network, they had done their collation and they boiled it down to a nice sort of manageable number of nodes, 383 nodes, right? And around 6,000 links which connect these nodes, right? So this is what the adjacency matrix would look like. Okay, so it's, you can say it's if I remember correctly, I think we had found the modular structure. I'll come to the how do you find the modular structure, but I think we had found the modular structure on this, and which is why you can see some sort of like some kind of structure along the diagonal. But anyway, so this is what that network looks like. And so we said, all right, so what we're going to do is to reduce any ambiguity, or to remove any ambiguity. What we're going to do is we're going to just remove any node that has a child, right? Basically any node which can be subdivided further, and we're going to remove any link associated with that node. Can that lead to a loss of information? Yes, and it probably did lead to a loss of some information, but we felt that the loss of some information is a price worth paying for the gaining of the fact that we now have no ambiguity. So now this is a completely unambiguous network, and now all the nodes are completely yes. Sorry, what?
What did exactly you do?
Okay, so going back to this network over here, right? So you notice that you see these guys over here, you cannot divide them. I mean, they have no subdivisions any further, right? Whereas this cortex has another subdivision and so on. We only just kept those nodes that have no further subdivisions, because you see these links are not just links between these guys. They're also links between the parents and the parents and the parents, the parents and the parents as well.
Except, for example, if I look at that pink section over there,
yeah,
there's a central node, there's a hub, right? Yeah,
yes.
So you would spread out, like remove that, map it, take one-to-one map it.
No, so we okay, so we do not keep those links anymore. So the diencephalon, we just remove all those links, because we were thinking of what we could do is we could just inherit the links from the parents. This is another thing that we said, let's just inherit all the links from the parents. But the problem is that didn't necessarily solve the problem, because you could be inheriting a redundant link.
So the entire links which are coming from diencephalon would be removed.
Yes. Because we only cared about those links between between those nodes which had no further subdivisions, right? Because these as I said, the links the diencephalon contains regions. It contains these regions. So a link from diencephalon doesn't mean anything distinct from a link between these nodes. It's just saying that at the scale at which we were doing this experiment, we didn't care to sort of identify which is the region within the diencephalon from which this link emanates. You get my point. It's not like it's emanating from some meta region. It's emanating from somewhere within the diencephalon. It's just that we don't know where.
So you remove that leaf.
So we remove the link in the hope that many of those links that we remove might have already been identified at the lower levels. Do you get it? So we say that we trust the links at the lowest levels because those links are at the finest level of resolution, right? These links are at a very poor level of resolution, and in any case, it means that this link means that there is a connection between some node in the diencephalon and some node over here. We don't know which ones, right? So that is the what we're doing over here.
Within most of the smaller nodes are converging towards higher regions, right?
That's okay. This might give that impression, but I think it's better to look at this way. This is, I think, the better picture to look at. So here you can see that we have removed this, and now essentially the basic structure is preserved, right? The basic structure of this has been preserved, and as I'll show in a second, not just that, but the degree distribution has also been preserved. So as I was saying, the loss of some connectivity information we feel is worth it, because essentially for the type of analysis that we wanted to perform, if we had the regions above, we can't do the analysis. It's not possible to do our analysis because it's ambiguous. We don't know. We're saying that there's a link between some level, some node within the diencephalon, for example, to some other node somewhere. We don't know which node it is. How is it going to help us? And it's not unique as well, right? Or we could already have the link, but we're just not even aware of it. So all these problems can arise. So as I said, so basically upon doing this procedure, we go from this network to a network where we have reduced the number of nodes, right? By not a huge amount, but yeah, it's gone from 383 to 266, but the links have dropped off a fair bit. But as we'll see, the macroscopic properties are quite similar, right? The other thing that I just want to mention before leaving this slide is that these brain regions, we did an independent sort of analysis of each of the brain regions in this what we call the leaf node network. We say these are the leaf nodes because we say that if you consider like a tree with branches, right at the very end you just had the leaves, right? What we have remaining are just the leaves. So we call this the leaf node network, and we found that of the regions that existed in this network, they range they vary a lot in terms of its volume. So it goes from around 2 cubic millimeters to around 2000 cubic millimeters. So there's a huge range in terms of the volumes of these regions. And this is another problem by the way. So suppose you have some of the regions in the original network, the volumes, I mean, if you have a huge region which has further children, and you don't know where it's emanating from, it's a huge problem. But at least now we know, okay, even though this is a large region, there are no further subdivisions. So it has to be, I mean, so at least there might at a later point in time come a subdivision, but at least with the information that we have, we know for a fact that this particular region has a link to something else. So it allows us to make our assertions very unambiguously. So hopefully I've convinced you with regards to this, because yeah, so I mean it is important. It's important because this is like the key sort of step that we made to kind of clean up our data in order to do our analysis. So as promised, I'll show you that. So this is the original network of Moda and Singh, right? And what they, I mean, essentially all they've done here, so they just remove the directions, and you can see that if you plot the degree distribution, right? So, but here they just shown that the probability that the degree is more than some x. Okay, so they just shown it in a different way. It follows like a almost perfectly follows like an exponential distribution. Right? So this is actually by the way one of the recent things that I was mentioning in the previous lecture, that the degree distribution is something which is very important to consider, because suppose you have a binomial distribution, Poisson distribution, scale free distribution, and so on. It's all well and good, but the point is, if the actual empirical network has a degree distribution like this, then you're sort of not comparing the same thing with your null model. So it's so this is what they see, and what we find in our network is something very similar, right? So essentially, so here they have just removed the directions, and they got this plot. In our network, you can just separately look at the in degree, the out degree, and the total degree, and we see that although there is a slight deviation for the case of the in degree, the out degree and the total degree very neatly follow an exponential, right? So that's very comforting, that we haven't really lost the main sort of macroscopic features, especially the degree distribution, which is very important. We have not lost that information, right? So that gave us some confidence that we can go ahead with this network and do our analysis, right? All right, so that's what we did. So now I will come to by the way, some of the pictures are better than others. I like this one, but some of them, there's one at the end which I don't know if, okay, anyway, we'll cross that bridge when we get to it. Yes, from Playtime. Yeah. Modularity is something that I've talked about a lot up to this stage, and now let's get into the nitty gritty of how we actually go about finding the modular structure. All right. So this can be a bit tricky for some of you, so let's go through this a little bit carefully. Remember, finding the modules is a little bit of it's there is as I was saying before, there is not a simple way to identify what is the best possible or the most ideal partitioning of the network that is going to show the properties that the connections or the density within the partitions are much more than the density across the partitions, right? So that is it's a very difficult question to do. So in its place, people typically come up with approaches by which they can try to come up with like the best possible partition that comes close to capturing the modular nature of a given network, if so it exists, right? So we'll go through one such very famous approach, which is called the spectral Newman method, with this following few slides. So okay, so how do we quantify the extent to which the communities are segregated? So first of all, suppose we say that, okay, so you have a node I, it belongs to some community Gi, right? And then firstly, we know what we have to find out, what are the actual number of edges that exist between nodes of the same community, right? So I'm just going to say that Gi is some community. Now, what is Gi? I could say, okay, Gi could be nodes 1, 7, 9, 10, 4, whatever. I'm just going to call this my community 1. Community 2 could be something else, and so on. So then I basically I do some partitioning in such a way that I say that these are my communities. Okay, maybe my guess is very good, maybe my guess is not so good, but I'm going to make this this guess, and based on that, I'm going to find what is the actual number of edges between nodes of this community. That is just given by this. So what does this mean? Well, this is a delta function. So basically, when Gi is equal to Gj, that means that I and j are in the same module, then I'm going to consider this, otherwise I'm not. So in other words, I'm only going to consider those parts of the adjacency matrix where I and j are in the same module Gij, right? Simple. And I'm just doing dividing by half over here because I'm doing double counting. In the case of an undirected network, I'm doing double counting. Aij is equal to Aji. So I'm doing double counting. I'm going to divide by half. And for a directed network, I don't need to do that. So I mean, I'm not double counting. So okay, this part is hopefully extremely clear, right? This is very straightforward. Next, suppose the total number of links in the network is m, then one can imagine, so that's just the total number of links. I'm just going to say, I'm going to count them, it's going to be m, and then I can now imagine 2m stubs. Stubs are basically just the two ends of the link. Okay, so one stub here, and that just connects the links together. Or for the case of a directed network, it can have some in stubs and some out stubs. So basically M in stubs, M out stubs. So for example, let's look at this case over here. I'm just going to imagine that at the ends of each links, I'm just going to have some stubs, right? So basically a stub here and there and there and there. And for the case of a directed network, I'm going to have separate out stubs and in stubs. So the out stubs are at the base of the link, and the in stubs are at the end of the link, right? And so I'm just going to focus on the stubs. Okay, so on the blue ones here, and then the blue and the yellow ones over here. So suppose all of these links were random. Okay, so there is assuming that you have a given number of stubs ki per node. So I'm just so I say here these have 4 stubs, 1 stub, 2 stubs, and so on. But if all the links were just were at random, then the expected number of links between stubs of nodes I and j is just this, right? Hopefully that's clear. Or in the case of a directed network, it's just that k, because you look at the in degree and the out degree for, you know. So fine. Hopefully that part is fine. So but the point is basically it's just very simple. So basically I've now calculated the expected number. So I found the actual number of links over here. I've also found the expected number of links from the total possible stubs that exist. This tells me at random, suppose I just assign the links in, how many links can I expect to see. Having the actual number of links and the expected number of links, furthermore, the expected number of links between nodes of the same community, I just now have to do this as well. I basically say I have the delta function. So when I and j are the same community, I add them, otherwise, and so on for the director. So now I have these two measures. I have the expected number of links, I have the actual number of links, and then I just subtract them. I say that if I take the difference between the actual and the expected number of edges between nodes of the same group, this should be in the two types of networks. It's just this, right? So basically, I'm just going to subtract. I'm going to take this, and I'm just going to subtract this, and I get this. And all I've done is I've just scaled. I've divided by m. To m is the total number of links. I've just divided by that. That's what I've done. And the same thing I've done over here as well, right? For the case of a directed network. So what this measure is going to tell me what is like the excess number of links seen within a group than would be expected by pure chance, right? So for example, suppose my kj by 2m, if it turns out that upon doing this, it turns out to be 0, it means that on average, whatever links I am actually seeing are basically more or less what I expect to see, right? And so that means that essentially there is no modular structure, so to speak. I'm just doing it at random and I'm getting the same thing. Whereas suppose it turns out that when I do this measurement and then I find that Q is high, that means what I'm seeing is actually more likely than what I'm seeing by pure chance. So what I'm seeing, the connections within the group is more than what I'm seeing just by pure chance. And that measure is what we are going to use going forward, right? And so this is what's going to help us to identify modular structure. Okay, so having this definition, so we take this now as granted. We now go ahead and we say, all right, so suppose we can do this for a given partition. We can identify what the Q is, which is basically the measure of the excess number of links within groups. How do we basically figure out what is the best possible partition? So this is the subject of a very famous paper by Mark Newman, and this is a follow-up paper where he extended this to directed networks. Actually, the directed network form is the one that we use in our study, but yeah, so this is a method which is now has been referred to as the spectral Newman method, because it's so widely used, and this involves modularity maximization, right? So essentially what is being done is that one assumes that each node, so first of all, before we divide it into like a large number of communities, we have to ask the first question: can one at least find if two communities exist, right? One can at least ask the question, okay, can you at least say, okay, fine, in this entire network, do you find that at least amongst these nodes there's more connection, amongst these nodes there's more connection, and between them there's less connections? Can you at least ask that question? So to answer that question, we say, all right, let's just assume that each node I is in one of two groups, and introduce a column vector S which has just two values, +1 or -1, depending on which group it belongs to, right? So if you do this for the case of an undirected network, it turns out that, so remember, you had Q was defined over here, but now this delta you can actually just write it in this form over here. It's just using the properties of S. So basically when I and J are the same, either both +1 or both -1, this becomes a 1, so 2, so this cancels out, right? Yeah, and then you can show that by basically, so this will simplify to this, and then in turn you can now write this as a new matrix which is called B, okay? And as I'll mention it in the next slide, B is what we call the modularity matrix, right? So essentially all I've done, I mean, essentially we just introduced this new column vector S and just simplified Q to this form, right? So now the same thing can be written as, the S transpose times BS. That's it. It's the exact same definition as before, not change anything. So I've now written Q like this, and as shown in this other paper by Newman over here, you can do something very similar for the case of a directed network, because now you have to be a little careful, because here the in degree and out degree, there's a difference. So the simple way to get around that is to just assume to use the fact that since Q is a scalar, it's equal to its own transpose. So all you have to do is you just say, okay, since it's equal to its own transpose, I can just redefine Q to be equal to Q plus Q transpose by 2. But I mean, since Q equals Q transpose, this is axiomatically true, right? So what we do is basically say, okay, Q equals half of, and this is, by the way, this is the same as this, except remember, for a directed network, you don't have that extra factor of 2. That's why you have instead of 4m, you have a 2m over here. So you have this, and then this is the transpose, and you simplify that, and you get this form, right? So it's actually very similar to this in the end, except now instead of B, you just have B plus B transpose. So okay, great. So we've moved one step ahead. So instead of just simply having Q in the form mentioned in the previous here, we have now rewritten Q in a way that it is, instead of being just in terms of A and the Ki Kj, is now written in terms of B and S. So why is this helpful? So remember, but S has only two values, +1 and -1, and that's going to come in handy in just a second. So yes,
It's a scalar, right?
yes.
So why bother writing it this way?
Okay, I don't remember the exact details in this paper, but it just allows you to sort of redefine, because you can't use this form over here for Q. It's not going to work because of the inherent directionality, because B and B transpose are not the same, right? So you need to.
The value of Q should be same, right, from both the expression.
Yes, no, the value is, but this will be so, but this is not the same as one second, actually. When you do the calculation of okay, I remember. This comes the issue comes when you have to take the eigenvalues, which I come in a second. Yeah, so that's the reason. Okay. Because here this will be non-symmetric, whether this is symmetric. So taking the eigenvalues is going to make a big difference. Yeah. So okay, good to get this part of that. This will actually it cancels out, because if you notice, if you just take the +1 over here what you'll get is basically just this, right? So you take this, the summation of this over this term. So summation of ij over kj will give you an m, that will cancel out, and then from the other one you'll get I think another, and then and the summation of aij also is going to give you I think 2m. So you'll get a 2m and then an m, and so it'll all just cancel. You can work it out. So that's what happens. But good thing that you spotted that. Yeah. Okay, cool. So so yeah, so now, right, so you have the q, two definitions, right? So for the I mean the reason why I'm mentioning specifically both undirected and directed is because I think it's very important, at least for our work, that it's important to note the fact that, you know, especially in brain networks, they can have directionality. So it's important to keep this in mind. So but this is how it's done in general for an undirected network. Anyway, the point is, now we have redefined q. It's the same as before, but now just in terms of S and B. Okay, whereas okay, so what do we do now? So we say that, all right, so the issue is to maximize q, right? That's what we said, we decided from the beginning. We need to maximize q and find the partition that maximizes q. So now the q is going to improve if you have a partition that is, you know, the best possible. Yeah, that is the best possible, right? So how do we do that? So basically, so we have to, given some modularity matrix B, we need to find the S so that q is maximum. Now S here is just a +1 or a -1, and remember what we are identifying S as is basically the membership. Am I belonging to the left side or the right side, or the plus side or the minus side? How am I going to do the partitioning? So for example, I could just say S is like -1 and then all bunch of +1s. What does that mean? It means it's just I'm going to have one node on the one side and all the other nodes on the other side. That's not a very reasonable partition, but I could do that, right? That's a possible partition, but maybe that's not a very going to give me a very good q, right? Or I could have exactly half of them +1, exactly half of them -1. It might give me a q, but I have no idea like whether that q is optimal. So I have to choose my S vector in a smart way. Now the insight of Newman is that if you notice this form over here, you just take the transpose. It's basically, it reminds you of taking the eigenvalue, right? So and so maximizing q is like maximizing the eigenvalue in this expression. So what we have to do is we have to find the S, which basically so we have to find the eigenvalue, the eigenvector of the largest eigenvalue of B, right? But there's a small problem, which is that we can't just take the eigenvector of B, because this S is not just anything. S is actually specifically +1 and -1. So what we do is we say, all right, we will take the eigenvector and then just look at the signs. We'll say we're going to take find the eigenvalues and eigenvectors of B. Look at the largest eigenvalue, corresponding eigenvector, and just look at the signs, right? And then if we say, okay, the plus signs, I'm going to just say, okay, this corresponds to S equals +1. The minus signs corresponds to S equals -1. I'm going to just make that approximation, right? So this is going to give me an elementary partition of the network. It's not, I mean, it's getting as close as possible to maximizing the eigenvalue, right, in this expression, because again I have some constraints as how I can change S. So it turns out when you do this, it actually does a remarkably good job, and they show an example in this paper. He shows an example in this paper where he does this for some simple networks. And this can actually be refined by using what's called the Kernighan-Lin algorithm, wherein at each step what we do is that every node in the subdivided module, you check if just by swapping them from one module to the other, whether that's going to improve or reduce the Q. You check that. If it does, you keep the swap. If not, you discard the swap. And the reason you do this, by the way, is because this is not perfect, because you cannot find the, I mean, you do not have what's it called, the option to choose any S you want. S has to be +1 or -1. So you said, all right, so this is, since it's an approximation, I'll have to come as close as possible I can to the best partition by trying out an algorithm like this to just see, well, maybe can I do a little bit better by doing a bit of swapping. Again, this is not essential, it's not essential to do this, but it helps to get even closer to a better partition. So okay, yeah, so so before I get to the next slide, so this is as you can see, I mean, it doesn't involve too many steps. I mean, once you understand the logic, you realize, all right, it's not too hard to do. Essentially, all it boils down to is finding the modularity matrix, which can be done very easily once you know the adjacency matrix, the the in degrees, out degrees, so on. You can just find the modularity matrix and find the largest the eigenvector of the largest eigenvalue, and then there you go. You just do a bit of swapping with the Kernighan Lin. It takes a few steps to do all of this, but at the end of it, you have a partition, right? Now, another thing that I should mention is he also talks about this paper. So as of now, you've got two partitions, right? So this is a very nice job into dividing this into two. So okay, so I can divide this network into two. Well, you can subdivide this even further. You can say, all right, but I've got my two modules. Can I divide, let's say, module A and module B? Can I divide module A further? You check that, and then so you do the analysis strictly on module A and do strictly on module B, and then you do it recursively. You keep saying, okay, I can divide this. Okay, keep going up to the point where I reach one submodule that I cannot divide. Then I say, okay, right, this is done. Try the next submodule, and so on. Through this recursive procedure, I can now find how many of the modules that can exist in this network. Okay. So this is the entirety of the spectral Newman method. And here is just what we did found in our network. So remember, we have a leaf node network, and we did this analysis, and we used the basically the spectral Newman method, and this fairly sort of like colorful looking picture tells you essentially how the network is broken down in terms of modules. Right? So what we found is that this network has 5 modules. Now you may ask the question, but how do we know this? How can we be sure? All those questions will be answered in subsequent slides. But the point is, with just strictly application of the spectral Newman method reveals modules like this. All right. Okay, so you may also ask the question, okay, hang on, how did I plot it like this? How did I know where the brain regions are? How do I know why are the nodes of different size and so on? All of that, but the answer to all of that is Anand. So he had spent several months of work painstakingly going through the the literature, and actually in the supplementary material of this paper we have like a table where he actually lists out the stereotactic coordinates of like the centers of each of the brain regions, and also their estimated volume and so on. And so essentially what we've done over here is the brain regions are plotted centered around in basically in a 3D space, right? So in the stereotactic coordinate space. And these are just 3 different projections, as you probably would have inferred by now. These are just the same brain but 3 different projections, and the size is proportional to, I mean, it's scaled by the volume. It's not exactly proportional, but it's scaled by the volume. I think I forgot exactly what the scaling was, but yeah, but basically broadly speaking, larger size equals larger volume. And the links are colored in accordance with the source. So if a yellow link means it's coming from a yellow source, and so on. Right? And this is like the scale. So basically that much is basically 5 millimeters. So you can imagine roughly what are the what's the separation between brain regions and so on. Right? So yeah, so that is what we find. And so you can see. So having sort of said all that, what can we find? So actually, if you look at, for example, at this projection, you find something very interesting. You find that actually there's a very interesting sort of spatial segregation of modules. You find that like towards like the back is mostly purple, here's mostly blue, mostly green, mostly red. The yellow one is a funny one. The yellow one seems to be sort of split into 2 like that. It's not even here. Sort of you can imagine a bit of red over here. There's a blue over here, and so on. But yes, but there does seem to be some sort of spatial localization of modules, which leads to a very natural question, which is that is this just simply happening because they're proximal, so they're just going to be connecting because there's two regions are close together, and so therefore just because they're close by they connect. Now we had that very same thought, and so what we did, and I'll talk about it in a bit later in the talk, is that we revised this method by taking into account the distance between the nodes when computing Q, right? So we now computed Q in a way that we accounted for the fact that, well, two nodes are a certain distance apart, so that should affect the likelihood of connection. So in other words, nullify the effect of distance. And not to spoil the story, but it turns out that this is not happening just because of proximity. So more on that later. But anyway, so as of now, so this is the observation. So all we've done, we've just taken the original paper from the network of Moth and Singh. We did some cleaning up based on what I had told you, the logic that I presented to you. We used the spectral Newman method. Of course, Anand spent months collating this data, and then we did this. And so these, by the way, these are just this is just like a list of all the the abbreviations of the various brain regions that are located in the different modules. You may ask the question, well, is there any sort of commonality between the region that we see over here in terms of their modalities and so on? And sure enough, that's something that we have found. And so, but before we get to that, let's just first focus on one particular thing, which is that, all right, so we have 5 modules. Okay, how are these modules organized in terms of the main sort of like lobes of the brain, right? So remember, we have removed or we have reduced the network to actually remove like the very high hierarchical level nodes, such as like the frontal lobe, parietal lobe, and so on. So we're only keeping those at the leaf node, but those at the leaf layer are in some sense children of those at the very high level. So in some there still remain a lot of leaf nodes that belong to the frontal lobe, that belong to the parietal lobe, and so on. So there is representation of all these lobes, right? Because those subdivisions still exist in a net. We of course haven't discarded them. So the question now becomes, what is the split of these different modules in terms of the different lobes of the brain? Interestingly, what we find is that there are some modules which, for example, you look at the basal ganglia, it's almost entirely dominated by nodes of module 3, just with a few others, but almost entirely by nodes of module 3, as you can see over here in this what's called an alluvial diagram, right? So basically this sort of gives you like a split up in terms of all the nodes belonging to these modules and all the nodes that are children of these different brain regions. So the basal ganglia, as I said, mostly belongs to module 3. The occipital lobe, interestingly, mostly module 5. Now already, by the way, that's giving you an indication of what module 5 might be doing. The temporal lobe is a bit of 3, a bit of 5, and a bit of 4, right? Actually quite a lot of 4, and so on. And so one can do the same thing for other ones. So for example, let's look at the thalamus. Actually it's a much more kind of localized region, so it's a bit harder to see here, but it's got a fair split of the different types of modules, basically 5, 4, 1, and 2. Whereas the parietal lobe is mostly 2, and so on. Anyway, but this is just to give the idea that it looks like that there are some brain regions which seem to be dominated by some modules, and some others which are some sort of mixtures of the two. So what does that mean? I mean, is that useful information? Now, well, we explored this further. So what we said was, all right, so let us look at all the regions that belong to, so basically go back to this table over here, right? this whole thing. So we went through all of them, or at least many of them, maybe not every one of them, but we went through many of these regions, and we said, what are these brain regions known for, or what is the known sort of functional role of these brain regions? Is there any kind of commonality? Right? So for example, this is from the paper. The nodes belong to module 5. I mean, and this is just the breakdown. So we said, okay, fine. So this picture over here, the first column is basically just so remember 5. There's a big little bit in the olfactory. There's a bit in the temporal lobe, a bit in the thalamus, and so on, right? So basically, that's what we're shown over here, and these are the corresponding subdivisions and so on. So it has that information, and what we've written down over here is some known functions of some nodes that belong to these regions. So interestingly, for example, for module number 5, you notice something very interesting. There's a lot of functionality associated with vision, right? So vision information relay, visual cortex, ventral visual pathway, and so on, right? It appears that there's some sort of sensory modality which is localized in module number 5. Module number 5 seems to be visual, and we did the same thing for the other modules, module 1, 2, and 4, and we said, okay, module 1 seems to be strongly associated with like the olfactory or gustatory senses, and then module 2 somatosensory, module 4 auditory, and so on. And these are just some examples of some some regions found over there. That's very interesting. So it looks like the modules are actually they correspond to the processing of some sensory information, but module 3 does not. However, module 3 is the only one which does not have any primary or sensory secondary areas, but contains the amygdala, which regulates emotional responses and so on. So we posit that could be having some different role from the other ones which are more associated with like sensory information, right? So already we found something interesting, right? So basically by just looking at how the modules have organized, it appears that the connectivity is revealing something about how the processing of information is organized, right? Okay, since I only have let me finish this and then we'll stop for today. So as I was mentioning numerous times, the partitioning that we come up with it's always going to be a question. Okay, have I found the best possible partition, right? I mean, so even the one with the spectral limit method does a very good job, but you can always say that, yeah, I mean, but maybe I could have come up with another partitioning that it could have had a higher Q value, for example, right? To answer that very specific question, what we did was we used this technique, which actually is sort of explained in nice detail in this paper over here, and you can check out this website as well for more information, which is that we used a simulated annealing technique wherein we basically start with some random partition, okay, and then at each step we do one of a couple of things. We either move a random node to a different module. So again, the modules already exist, right? But these are just chosen at random. So this is not a good partition. We just start with some partition, but we just move a node to a different module, or we merge two modules, because again, starting with random, we can of course have the freedom to merge two modules, or we can also split the two modules to try to minimize the connections between them, right? So we can, so these are some steps that you can do, and if the new partition leads to some change delta q, if delta q is negative, which means it's actually become worse, we can still accept it with some probability given by exponential of minus delta q by t, but t is some temperature that we have assigned at the beginning. So we started with some high temperature to say that, okay, initially we can allow things like this to happen. We can allow, even if delta q is negative, we can allow it with some probability, okay. but then over time, what we do is we use a cooling schedule, so that over time the delta t is going, the t is going to get lower and lower. So then at larger times we are not going to accept these sort of changes if they're negative. But if the delta q is positive, we just accept it. So okay, it's improved the mortality, fantastic, I'm going to accept it. so if only that delta q is negative, then we have to think about it. But delta q is positive, we immediately just accept it. So we do this, right, and we do this over like 1,000 realizations, and we come up with a large number of possible modular organizations. It's the same network, but a large number of possible modular organizations. Now we have an empirical one which we got for the spectral Newman method, right? So how does it do compared to that? Very interestingly, the spectral Newman does as good, if not better, than almost all of them. Right? So this picture is a little tricky to explain. I mean, again, you can check the details over here. Essentially, what they've done is they just done some projection using curvilinear coordinate analysis. But the x and y axis are not what's important. What is important is the z axis. Okay, so that's the key over here. The z axis is telling you what is the modularity of your partition, right? So you notice that you have a large number of guys down here, but having a very low modularity, which is it's not found a good sort of partition, and maybe just found some local minima and just got stuck there, right? But up here you can see that there's a large number of them that's come pretty close to the q value that we have, right? You might have some which is maybe slightly better, but not really significantly so. In fact, what we find is that there's around 291 of the partitions are within 3% of our empirical value, right? So our empirical value is pretty good, which means the partitioning that we got is actually, it's actually we can really trust that it's good. It's close to as good as you can get. Basically, that's the point, right? So this was actually very reassuring for us, right? Okay, so yeah, maybe I'll finish this slide and then we'll stop, because I think there's got a lot more to cover tomorrow. So so the other thing is, of these 1,000 realizations, so we say okay, fine. So some of them have done really well, right? So but how likely are we going to see like the same structure happening again and again? Because you got similar q values, but what about the actual organization, right? So in other words, just because the q values are the same doesn't mean that the organization, I mean, I could have a very different sort of partitioning which give me the same q value, but I want to check how close is my partitioning. For that, what we do is we use what's called the consensus matrix, where Pij is the probability that a pair of nodes are found in the same module, right? And this is done over the 1,000 realizations, and we see that, for example, this module 5 is almost completely consistent across partitions, which means that no matter which partition you do, module 5 is showing the same set of nodes that keep co-occurring together again and again and again, right? And many of the others also show a fair degree of stability as well, this is also very, very good to know, because not only is our q value close to as good as you can get, but also the partitioning that we have is fairly consistent with what you would get over a whole sort of ensemble of partitions that you get using simulated annealing, right? And what we see over here is just simply another way of looking at this data, which is that like what is the what's it called, the versatility of a given node, and we want nodes to have as less versatility as possible, because we don't want them to keep switching around between modules. And what we see, for example, is that you see most of module 5 has a very low versatility, which means that it's almost never changing. So basically it's defined like this. So Vi is the sum of sine pi Pij by the sum of Pij, and as I mean, you can work out that when this is going to be the highest when Pij is 0.5 for all ij, and lowest when it's either 0 or 1, right? So when if Pij is going to be close to 1, it's going to lower your versatility, or close to 0, of course, also going to lower your versatility. And of course, in the nodes in module 5 have very low versatility, which is good. In fact, as you can see here, close to around 200 nodes have fairly low versatility, then it sort of jumps up over there. So these nodes have a bit less versatility. So this is around like 70% or so of the nodes. So what we mentioned up here, 70% of the nodes have low versatility, which means that they are more or less occurring in the same partitioning again and again. And this is actually a very, very strong statement, because now we can say that everything that we have said so far about, you know, the sensory modalities and so on, we said, okay, somebody could argue, okay, but you just did this for a single partition, how can you be sure about this? But I said, well, look, this is actually showing that regardless of the kind of partitioning that we're using, the same nodes more or less are occurring together, right? And this is further sort of proof that our observations regarding sensory modalities is something that is actually a genuine structural feature of the network, not just something that's happened by chance by our particular method that we've used. Okay, so okay, exactly 5 o'clock now. So hopefully, yeah, I think I can finish this tomorrow. So I think with that, let's call it a day. And if we have any further questions, we can ask. Otherwise, we'll talk tomorrow.